All right, we are now on lab 6 out of 8, so we're getting there. We're past halfway already for the semester. This particular lab is for protein unfolding. The protein in question is myoglobin. So we're not really interested in the protein itself. We're interested in the techniques and in the thermodynamics that goes with it and in instrumental methods. What we're going to do is the following. So first of all, myoglobin is an O2 carrier in skeletal muscle. So we have it inside of us. Most of the O2 carrying business is done by hemoglobin, of course. It's a more efficient molecule, but O2 is present in the skeletal muscles. Apparently nobody seems to be real sure what it's doing there. You can knock it out in mice, for example, and the mice still live, so it isn't utterly essential. But it seems to be that it's a pool of stuff that can pick up oxygen in the muscles and give them an extra load of oxygen that they can store right there, right nearby. And that's an advantage, of course, to your muscles, which need a lot of oxygen if you're running or something. All right, so as you raise the concentration of oxygen in the environment, the pressure of oxygen, partial pressure of oxygen in the air, let's say, then myoglobin binds to O2. As you reduce the concentration of O2 in the environment, then myoglobin releases O2. And of course, once you've loaded it up, it's the release of O2 into the tissues that both hemoglobin and myoglobin are good for. But all of that is the biology. What we're interested in is the thermodynamics, the physical chemistry. We're going to denature or unfold the myoglobin, and then we're going to try and measure the free energy. We're going to do that using fluorescence spectroscopy and an unfolding or denaturing agent called guanidinium. So here's the overall reaction. You have myoglobin in the folded state. It goes to myoglobin in the unfolded state. Capital F for folded, capital U for unfolded. There's a Gibbs free energy change associated with that. This is the quantity we're after. To get that, we're going to measure the fraction of the myoglobin that is in the unfolded state at any one point under any one set of conditions, under a given denaturant concentration, as it turns out. And then once we have that fraction, we can use that to get the equilibrium constant. We'll see how that works. The equilibrium constant is the ratio, not the fraction, of unfolded myoglobin to folded myoglobin. And that is related directly to the Gibbs free energy. So clearly, if we can measure these guys, or if we can measure the fraction, it turns out, then we can also get the delta G. That's the gist of it. But there's a little bit of a hitch. Myoglobin's a stable molecule, doesn't unfold at room temperature. We could heat things up and unfold it with temperature, and I think there are experiments that do that, but we're not going to do that. We're going to use, and in fact, the way we're going to do it is the standard way that people do it everywhere when looking at protein folding and protein unfolding experiments. We're going to use a denaturant, something called guanidinium. That's this stuff here, which has the property that if you put it in solution with the myoglobin at high enough concentrations, and we're going to go to 8 molar at one point here, so we're definitely going to get to very high concentrations. If you go to high enough concentrations anyway, it destabilizes the protein. Probably, proteins are held together by hydrophobic interactions, and I think this molecule just disrupts those hydrophobic interactions. It basically binds to the hydrophobic regions better than the protein itself does, at high concentrations anyway. And so I think that ends up unfolding the protein, but it does so as a function of concentration, right? If you're at low concentration, it doesn't unfold the protein, or it mostly doesn't unfold the protein. And as you raise the concentration, more and more of the protein unfolds. We can measure that, measure the fraction that's unfolded at every point, at every concentration, and therefore we can get the free energy as a function of concentration, and that's what we're after. So here is our reaction again. F goes to U, and as a function of guanidinium concentration. the quantity that's really interesting, that's of biological and physical significance, isn't the delta G, though, at any one concentration of guanidinium. After all, that concentration is kind of an artificial thing. We're sticking that in order to melt the stuff. What we want is the delta G at pure solvent or pure phosphate buffer. I say water here, but it's really phosphate buffer. So we want it when the concentration of guanidinium is 0. We're going to extrapolate to 0 in order to get that. All right, so how are we going to go about doing this? Well, we're going to use fluorescence to measure this fraction, and I'll talk a little bit below about how the fluorescence works, but basically the fluorescence intensity that you measure depends on how folded or unfolded the protein is. And so we can define this quantity, the fraction that's unfolded, in the following way. We measure the intensity at one concentration. We measure the intensity when it's completely unfolded. That's at the maximum concentration when presumably there's no more unfolding to happen. And then we divide that difference, we take the difference between those two, and we divide that difference by the total range of possible fluorescences, which is the full unfolded fluorescence over the full folded fluorescence, minus the full folded fluorescence. And so this is a quantity that ranges between 0 and 1 as the protein unfolds, and in fact is related to is in fact the fraction of the unfolded protein. If, that is, this is true, or this relationship holds, if the protein unfolds in a single step. In other words, if you go from a fully folded protein to a fully unfolded protein with no intermediates in between. You have stuff like this happening, it partially unfolds, and then it unfolds a little bit more, and then it unfolds still a little bit more after that. Then this relationship won't be good, and our whole analysis will be bad. And so if we get bad results at the end, and we got bad results last year, so I'm not sure that we won't get bad results this year too. If we get bad results, then the fact that there may be intermediates between the folded and the unfolded state, the fully folded and the fully unfolded state, may be the reason. At least that's a good thing to put in your write-up, maybe, if you don't get good results. All right, so how does this work? How does this F give us what we want? Well, the fraction unfolded is the following, right? It's the amount that's unfolded, the concentration that's unfolded, over the total concentration of the whole protein, that is the unfolded plus the folded stuff. That's obviously the fraction that's unfolded. If I divide top and bottom by the concentration of unfolded stuff, then this fraction is 1 over 1 plus the ratio of folded to unfolded. But according to our reaction here, F goes to U with an equilibrium constant. That equilibrium constant would be U over F, right? And that means F over U here just has to be 1 over the equilibrium constant. And so the fraction here is related to the equilibrium constant, which is related to the Gibbs free energy. So, and that's of course related to this fluorescence measurement that we're making. So we're going to be basically measuring something that's directly related to the Gibbs free energy change we want when we measure stuff with our fluorimeter. All right, solving for delta G. So we can solve this, right? This relationship for delta G. If you do that, you get this, that the delta G at each and every concentration, right? This fraction will vary as we change the concentration, and therefore we'll get a different delta G at each concentration, because after all, the free energy of unfolding like this depends on what that concentration is. If the concentration's high, the free energy of unfolding is a lot more favorable than if the concentration's low. So this delta G should depend on the concentration, and we'll get that at every value of the concentration that we use by just plugging in this measured number we get from our fluorimeter, right? This is the experimentally measurable quantity. Okay, in order to get the concentration at the delta G at concentration 0, we need some sort of relationship between delta G here at any concentration and delta G at 0 concentration, and that's this relationship. We assume a linear relationship between the two. How reasonable is that? Well, experimentally it seems to be true. If you plot delta G versus the concentration, you get a straight line, so it seems to be a good relationship. What the basis for that is, I don't know. I haven't thought hard enough about it to know whether that's something that sounds reasonable. I think it's an approximation for something else. I think this should really be log of 1 minus C or something like that. It really shouldn't be linear in concentration by everything I know from stat mech and thermo. But all right, if it works, and that's good enough for us for now. All right, so we can do two things then. We're going to analyze our data in two different ways. We'll of course talk more about the analysis next week, but for this week we'll say just a little bit about it. Here's one relationship, right? We have the fraction of stuff that's unfolded as a function of delta G, this delta G0, this is the quantity we want. And we'll then get, and then we have another adjustable parameter, which is this slope, right? This is the thing that gives us how quickly the delta G changes when I change the concentration. So it's a slope quantity. That's why it's called M here. So we've got two adjustable parameters. And so we can plot the, as a function of concentration for various different concentrations, we can plot the fraction that's unfolded. And then we can fit that curve by varying, in solver or something similar, by varying the quantity delta G0 and also varying this slope as adjustable parameters. So that's one approach. And the second approach is that instead of plotting the fraction itself, we solve for delta G. We use this relationship, and we just plot delta G as a function of concentration, and then we should get a straight line, right? According to this relationship here, delta G versus concentration should be a straight line. The intercept should be the quantity we want, and the slope, that M parameter, should be the negative of the slope. And so by taking the slope value, we get an estimate for M, and by looking at the intercept, we get an estimate for G0. Now, when you do things like this, usually the slope is pretty good. Usually the intercept is really bad. And that's because any error in the slope here, amplified and levered out till you get to 0 concentration here, makes a great big error. It's easy to get a great big error in that intercept. And so I suspect that this method up here will give us better results, but we will see. All right, so two different ways of analyzing the data should give similar results at least. And again, we'll talk about that more next week. Next thing I want to talk about is the actual fluorescence process itself. So I think everybody knows this kind of stuff already. This is a Jablonski diagram. Probably, I hope everybody has seen these things before. Here's the excited state energy surface. Here's the ground state energy surface. There's a manifold of vibrational states on both surfaces. And what happens is, usually you're in the ground state vibrational state. You send in a photon, a high energy photon, a blue photon, as I've drawn it here. And that causes a transition, an electronic transition from the ground state up to the first excited electronic state. But it usually doesn't go to the ground vibrational state, which means that it's got some extra vibrational energy after the transition. And that very quickly decays down. Basically, all that extra vibrational energy gets put out as heat into the surroundings, into the molecules around the absorber. And you end up in the ground vibrational state, even if you didn't go there originally. But that means that you've lost some energy to the surroundings already. So after that, the electronic excited state emits a photon. That's the fluorescent photon. There's usually only about a 1 nanosecond delay. It's very, you absorb, a nanosecond later you emit. So it's very quick, the fluorescence process is. But that fluorescent photon will be at lower energy, so I drew it red here. And because it's at lower energy, you can put filters in. So you're sending in some excitation energy at high energy, in our case about 300 nanometers. 285, 296, I think, is what the write-up says. And I don't think it matters a great deal what exactly the excitation energy is. It's got to be within the envelope of the absorber, which in this case are going to be tryptophans on the protein molecule. But then the emission comes out at a different and longer wavelength, which means you can put a filter in here, not see the excitation, only see the emission, and it looks like the molecule is glowing. For all intents and purposes, the molecule is glowing for us. All right, so that's how the fluorescence process works. But what good does that do us? Well, okay, so before we go to what good it does us, what is it that's actually fluorescing in the protein? Well, there's a bunch of fluorescent residues naturally occurring in the protein. They all have a quantum yield, right? The quantum yield is how many fluorescent photons do you get out per incident absorption photon. And a lot of fluorophores have really bad quantum yields. This guy has a really bad quantum yield. That guy has a really bad quantum yield. This guy doesn't have such a great quantum yield. I forget what it is. It's like a few percent, I think. So that means only a few percent of the photons you absorb actually fluoresce. The rest of them decay out as heat into the surroundings. But even so, it's the best one. So most of our fluorescence is going to come from the tryptophans in myoglobin, and there are a couple. I think there's only two. All right, so what good does that do us? Well, if your protein is folded up like this, and then you've got these two green guys, those are the two tryptophans, well, they're next to a heme. The myoglobin has a heme. It's the heme that actually binds the oxygen. It's just like hemoglobin. And the heme is a good chromophore, and its absorption bands overlap with the emission bands of the tryptophan residues, which means that while it's folded up and they're close to each other like this, all the light that gets absorbed by the tryptophans ends up getting funneled over into the heme, and then it gets dissipated as heat, so they don't fluoresce. But if you open the thing up, the heme is released, or at least it's not as close to the tryptophans as it was, and so now the tryptophans can start fluorescing. And so what you see is as the protein unfolds, the fluorescence actually increases. So we're looking for an increase in fluorescence as we denature, as we increase the denaturant concentration. Going back up here for a second, ooh, lots of pink. Here we are. Here's our measure, right, or the thing we're going to actually look for, the fraction unfolded. The fact that the fluorescence is higher in the unfolded state means that this intensity, fluorescence intensity, will be bigger than that fluorescence intensity. And so when the intensity starts to approach the intensity of the unfolded state, then this fraction will go to 1, right? Because this will become IU almost. On the other hand, if we're back at the beginning of the process and almost nothing's unfolded, then these two guys will be almost the same. We'll get a 0 up on top, and the fraction will be basically 0. So it all makes sense. All right, back to where we were. Okay, so there's the unfolding. Here's what's called the fluorescence resonance energy transfer FRET process, and there's lots to say about this. This is called the Forster mechanism. It's the basis for a whole lot of biophysics. It's really very interesting and useful, but we won't talk about that too much this week. Maybe I'll talk about it next week a little bit, just as an aside, an interesting bit of physical chemistry on the side. It isn't real central to this lab. The only thing we really need to know is that the fluorescence does indeed increase when the protein unfolds. We don't need to know the details of the physical mechanism, but this is likely to be, I'm not even sure, but very likely this is the physical mechanism by which it happens. Basically, you've got a fluorophore here, and its emission band overlaps with the absorption band of a nearby chromophore. In our case, that would be the heme mainly. And so basically, as the fluorophore tries to emit, instead the photon just gets absorbed by the nearby chromophore. Now, it isn't quite emission and absorption in the normal sense because it's really dipole interactions here. That's what the Forster mechanism is all about. But in essence, it's emission and reabsorption by close-lying chromophores and fluorophores within the single molecule itself. All right, so that's the sort of theory and background and goals and all. What's the actual experiment? We're going to make 22 samples and collect 22 different fluorescence spectra, each one at a different guanidinium concentration. So we're going to set up a whole bunch of samples, each one at a different concentration, and then we're going to just take the spectrum of each one. So the whole experiment is setting up the samples and taking a bunch of fluorescence spectra. There's a table for how to set up those samples in the write-up. Let me say something right now because it's important. The write-up is for hemoglobin, but I switched to myoglobin this semester. I hope that doesn't cause trouble. I haven't tested it, I confess. I'm assuming that myoglobin will behave pretty much the same as hemoglobin. I switched to myoglobin because hemoglobin is a tetramer of myoglobins, basically. Each of these ones is basically a myo, and hemoglobin is the whole thing. And last year we got really bad results. We were off by more than a factor of 10 in the delta G that we should have gotten. I think that's because the thing we were actually measuring wasn't the unfolding of the myoglobin subunits themselves. It was the busting apart of this tetramer. So I think the tetramer busted apart. That caused a fluorescence rise by itself, and we never saw the unfolding of the actual myoglobin subunits. So by switching to myoglobin, I'm hoping that we get better results this time, but there is some risk that it won't work at all. I hope that doesn't happen. I'm going to come in on Tuesday and check it out myself, make sure that things seem to be working okay, and there may be some last-minute changes if things aren't working okay, just to warn everybody. All right, so what we're going to do is set up these 22 samples, each at a different guanidinium concentration. We're going to put them in a water rack at 25 degrees, and we're going to let them sit for an hour. Why? Because it takes that long for the unfolding process to occur. Unfolding doesn't occur instantly, so you got to let them sit for a while. Sitting for an hour means we're twiddling our thumbs for an hour, and of course we're always kind of running short of time in these experiments, so this is a relatively long experiment and we should be efficient. So everybody should be highly prepared to work efficiently. If you spend an hour setting up these vials and then you got to wait another hour and then you got to wait some more because there's only one fluorimeter and the other guys are in front of you, well, you might be there late and you don't want to be there late, so be efficient. All right, and then we're going to, once we've got everything ready, once the proteins are unfolded in each of the samples, so these are in little plastic tubes, all in a rack with little lids on them and everything. And you take a 3 milliliter aliquot out of that and put it in the spectrometer and the fluorimeter and just take a spectrum and, of course, collect and save that spectrum every time as soon as you get it. Don't let it sit on the machine. But apart from that, everything, that's it. All right. So we're going to make plots like we did, like I showed up above. Let's see, like either this one or this one. And then from those, we're going to determine the value of delta G0 and the value of the slope. The value of the slope isn't of great interest, but the value of the delta G0 is, and that's out there in the literature. It's, I guess I'll tell you, it's something like 45 kilojoules per mole. Last year we got something like 6 kilojoules or 7 kilojoules per mole. And that was, me and Amy got that, and everybody else got that too, or something along those lines. I think it was ranging from around 6 to 10 kilojoules per mole, so way off. And I'm hoping the switch to myoglobin is in the hope that we get more reasonable values this time. And then whatever you get, good or bad, of course, a write-up that explains the results. A couple of notes before we quit. First, the phosphate buffer that gets a fair amount of write-up space in the instructions is already prepared for you. You don't need to do that. Making a phosphate buffer is already a lot of work. We don't have to do that. And, not very exciting or very instructive, so yes, just take the phosphate buffer as it's given. I should say that phosphate buffers are a pain enough to work that they're routinely and widely bought pre-made in all labs, all chemical labs, all biochemical labs these days. I don't know if anybody ever makes a phosphate buffer from scratch anymore. And so that's what we're doing too. Guanidinium solution also has already been made, so that also will save us some time. This just says, yes, we're using myoglobin, not hemoglobin. The big thing about that is, since hemoglobin has 4 myoglobins, basically, I mean, they're not really myoglobins, but something like myoglobins inside them, it's basically got a molecular weight that's 4 times bigger than the molecular weight of myoglobin, which means if you're making a 20 micromolar solution for your stock solution of myoglobin, then you have to put in a different number of moles, right, of myoglobin because, well, a different weight of myoglobin because the molecular weight's different. So for horse myoglobin, the molecular weight is 16,951. I think that's what we have, but I have to go check the bottle. So I'm not entirely sure that's the exact molecular weight, but it doesn't matter. Molecular weight doesn't change that much from one species to another. It's around 17,000 for all species. The concentration that you're aiming for should be 1 milligram per milliliter or thereabouts. It doesn't matter exactly which concentration you get, as long as you know that concentration. You do need to know the concentration, of course, when you're doing your experiment. And it has to be high enough, right? The reason for this particular choice is so that there's enough stuff in the solution so the fluorimeter gets a good signal. So you don't want it to be too much lower than this, or too much higher for that matter. And also, in the write-up, you'll see 2 tables for how to prepare your solutions, your vials, with their concentrations of guanidinium. Guanidinium. One is labeled hemo and one is labeled myo. Even though we're using myoglobin, use the hemo table. The myoglobin table is for a different type of experiment altogether. It's called myoglobin, but it's not for this kind of experiment. So use the hemoglobin table. The table is in terms of actual volumes of stock solution. So as long as our concentrations of stock solution are okay, doesn't matter whether that's a stock solution of hemoglobin or of myoglobin. All right, and I think that's it, unless there are questions.